All right. Good evening, everyone, and welcome to tonight's Susquehanna University Alumni Lecture. My name is Kathy Straub, and I serve as Dean of the School of Natural and Social Sciences at Susquehanna University. I'm so pleased that you're joining us tonight for a lecture by Dr. Dave Richard of the Department of Biology. By way of background, Dr. Richard was born in London, and by the young age of 4, had already decided to become a biologist. Eventually, after grammar school and with degrees from Scotland, including a zoology PhD from Edinburgh University, he conducted research on insect development and taught undergraduate students at the University of North Carolina in Chapel Hill. He joined the Susquehanna University biology faculty in 1993 and was an early advocate that study away is a crucial piece of an undergraduate education and developed a long-standing trip to Australia. During the pandemic, he served as the university's COVID coordinator, where he oversaw and directed 2 years of our responses to the crisis. Dr. Richard has always been adventurous as a skydiver, a skier, a climber, and a scuba diver, as well as running marathons, until his knees rebelled. These interests led to the development of a course entitled Exercise and Extreme Physiology that in turn led to a Go Short trip based around a high-altitude trek in the high Himalayas. Dr. Richard was named Presidential Professor of Biology at Susquehanna in 2022. Sadly for us, he is retiring at the end of this academic year for a total of 31 years of service to Susquehanna. We wish Dave and his wife Allison the best as they begin the next phase of their adventure. Dave's talk this evening will discuss aspects of life in high altitude and other extremes, and will include some of his work with students over the years. Dave?
Thanks, Kathy, and welcome everybody. Thanks for coming along this evening. I'm going to spend a bit of time talking about 3 stories, all of which have to do with my work at various capacities with undergraduates. So let me just start by sharing my screen here. We'll try and run this off my iPad, so hopefully that'll work okay. So yeah, I'm going to talk a lot about the effects of hypoxia. Hypoxia being the lack of oxygen. And I'm going to spend a lot of time talking about work that I've done and time that I've spent, as Kathy says, up in the high Himalayas. This is one of my favorite mountains in the background here. This is a mountain called Ama Dablam. It's way out of my league as far as climbing is concerned. But when you go up to the Everest Base Camp region, you spend several days in the shadow of this beautiful mountain. So what I'm going to talk about is the 3 stories: the effect of altitude on climate change and hypoxia gene expression. You wouldn't necessarily think about putting those 2 things together. And this is based upon work that I've done with undergraduates in my research lab over the years. I'm going to spend some time talking about the cardiorespiratory function at high altitude in humans. And this has to do with the GO program that I lead up to the base camp at Everest, where we monitor students all the way up to make sure everybody's okay. And then finally, I'm going to talk a little bit about the diving reflex in humans. And this is a phenomenon that we still have as an evolutionary vestige. We're not as good at it as whales and seals and the like. And this reflects work that I did in the teaching lab in this class that Kathy mentioned, Exercise and Extreme Physiology. So starting off talking about climate change, and I apologize, Kathy, I know this gets into your area of expertise, so please forgive me if I make the odd mistake here and there. So just briefly, in the shadow of Nuptse here on the screen, I just want to briefly touch on where we are and some of our understanding about climate change. We know that climate change is largely the result, if not exclusively the result, of the release of greenhouse gases, one of the major ones being obviously carbon dioxide, and that's why we're all trying to decarbonize to the extent possible. And what the graph here is showing us is the historic carbon dioxide level over about the last 800,000 years. And you can see for most of that time, it sort of went up and down, somewhere between about 180 parts per million and 260 parts per million. And then I'm afraid my country back in the 1800s started developing the Industrial Revolution, and we started burning a lot of coal, and that caught on because, you know, it was a nice easy fuel, and oil and gas soon added to it. And so since the mid-19th century, we've been seeing a gradual and now explosive increase in carbon dioxide level. The number now is around about 420 parts per million, and it has never been above this line in hundreds of thousands of years. We can see this when we look at what's known as the Keeling curve. And this is a continuous measure with several readings taken each day at Mauna Loa. It's a University of California, San Diego venture. And what they've been doing is, since about 1959, is they have been plotting the daily and then for annual increases in carbon dioxide. And you can see it's a pretty inexorable rise here. If anything, it seems to be accelerating. It seems to be becoming a little bit exponential there. One of the interesting things about this, in my view, is this seasonal fluctuation. And so if we just break down one of the peaks here, so 1 year, January through December, what we see is an increase in CO2 levels in the winter and a slight decrease during the summer. And this reflects the fact that there's more land in the northern hemisphere and therefore more trees, more vegetation. And during the summer months, they take CO2 out of the atmosphere for photosynthesis. But nonetheless, you can see that there is this pretty drastic rise in CO2 levels within the atmosphere. Oops, too far. Now, in terms of, let me go back again. In terms of what this has done for temperature, and this is work, the left-hand panel here is work out of Michael Mann's lab at Penn. And what they've done here is they've plotted the temperature over the last 1,000 years or so. The ones in blue are indirect measurements from tree rings and such like. And then the data in red are direct measurements of temperature. And again, you can see that the temperature really rises drastically with the onset of the Industrial Revolution. This is the graph that's known as the hockey stick, and you can see why it looks like a hockey stick. So that's where we are right now, and where are we likely to get to? And so if we project forward, and this is the IPCC data here, we're already at this dotted line. We're already about 1 degree above the pre-industrial levels. And depending upon what we do, and the, as it were, political will that we may or may not display, we could end up with almost 2 degrees above, 3 degrees, or 2 and a bit degrees, or in worst case scenario, perhaps 4 degrees centigrade. This is all in centigrade, not Fahrenheit. 4 degrees centigrade above pre-industrial levels. And there's no way of sugarcoating that. That's just going to be catastrophic on a global level. Now, one of the things that's particularly interested me is there are some parts of the world that are particularly sensitive to climate change and to the overall warming. The poles, for example, the North Pole and the South Pole have been warming at a much faster rate than most of the planets. Likewise, the Himalayas, the high Himalayas up here, so the northern region of Nepal and the Tibetan Plateau in Tibet there. These have been rising in temperature much faster than lowland areas. And our little graph down here shows you the amount of temperature increase per decade based upon the elevation. And what you can see is at low elevation, you pretty much have a relatively low increase per decade. And as you go higher and higher in elevation, so this is up to 5 kilometers, 5,000 meters, which is about 16,000, 17,000 feet, you can see the rate of temperature increase is about 3-4 times what it is at the lowland area. So Himalayan regions, high mountain regions are particularly sensitive to climate change, to this global warming. And this can have a number of effects. Glaciers are being, eroded, are being destroyed and undermined at drastic rates and accelerating rates. The picture on the left here is an absolutely beautiful glacial lake called Gokyo Lake in the Everest region. And it's been filling up as there's been more and more glacial melt coming into that. And just the other side of the ridge here, you see the Gazompa Glacier. And when I walked across this last year, I took this little video of the amount of meltwater coming out of the glacier. This is on the glacier itself. So all this up here is ice that's covered with moraine. So these glaciers are not those sort of pristine white things that you would expect. They're mostly covered in ground up mountain on top of it. But yeah, there's been increased melting rates in the glaciers. There's also been decreased snowfall. This is the Lobuche Glacier, which is on the route up towards Everest. Before 2006, there was an average of about almost 1 meter of snow per year. 3 feet of snow per year. Since 2006, with a couple of odd anomalies, the snowfall's gone down to about 6 centimeters per year. And so you can imagine with increased melt and with decreased snowfall, the glaciers are retreating rapidly. This pyramid here is what's known as the Italian pyramid. It's a climate research station that used to be run by an Italian scientific organization. It's now been largely abandoned, which is rather a shame. And the guy who works here, Kazi Bista, he does almost daily measurements up on these glaciers, and he's the one who's produced all this data over the years. And when we go up there with students, we actually stay in the lodge that's in the basement of this pyramid structure. And coincidentally, it's where last time we were up with students, I came down with COVID. And so this was me saying goodbye to the students. They were going up the mountain, I was going down the mountain, and we caught up with each other a few days later. 16,000 feet is not an ideal place to have COVID, I can tell you that right now. 13 out of the 16 of us on the group actually came down with it, which was honestly not great. But nonetheless, everybody got through it just fine. Now, if you come out of the pyramid and head north, you come to the Khumbu Glacier. And the Khumbu Glacier is the one that leads up to Mount Everest. So this is the glacier here, and this is the famous Khumbu Icefall, through which the climbers have to go. And just peeking up above the ridge there's Mount Everest with the south summit just there. And so if you go up this, you get a really nice view of the glacier itself. And one of the disturbing things about this, and this is a picture I actually took back in 2011, this line here denotes the level of the glacier in 1952. And this line, when I took the photograph, denotes the level of the glacier in 2011. So you can see there's been quite a retreat in terms of the thickness of the glacier. The astounding thing to me is this is 300 feet. This is a massive drop in the volume of the glacier in this region. So what we have then is accelerating warming. We have loss of snowfall. We have increased melt and just general warming in the area. And that's having an effect upon the biology of the area. And what we're finding is, as it gets warmer at higher altitudes, insect species are migrating upwards. And this is really where I come into this story. And the photograph on the bottom left there, this is a butterfly that I photographed at about 15,000 feet a few years ago. And it shouldn't be there. I mean, this is almost in winter, and it was warm, and this butterfly was very happily flying around. We've seen flies and other insects up at base camp, and they really shouldn't be there. It should be too cold for them, in December especially, and it's not. So we're seeing an increase in invasive species, some of which are agricultural pests, some of which are disease vectors, such as mosquitoes. We're seeing more and more mosquitoes higher and higher in the Khumbu region. As an example of vertical migration of insects, we can look at these guys, Bactrocera, and it's a type of fruit fly, and it normally was found in this lowland region here, which is called the Terai. And the Terai is the southern border of Nepal, where it borders onto India. And it's very low, it's very humid, it's very warm. I mean, it's very beautiful, but it's not normally sort of a high altitude area. And these fruit flies, they would lay their larvae in oranges and other fruit. And you can see the larvae here. And, you know, they obviously spoil the fruit. It becomes unsaleable and largely uneatable. I should say this work here has been done by a colleague, Debraj Adhikari, who works out of university in Kathmandu. And what he's found is that over the last few years, we're now finding these larvae in Kathmandu itself. And Kathmandu is about 4,500 feet above sea level. So in the last few years, these insects have migrated up over 4,000 feet. And this is just one example. And they're continuing to move up higher and higher. All right, so what effect does that have upon the sort of the biology of the system? So just to sort of bring you up to speed here, what you find is as you climb in altitude, the overall pressure of the air decreases. And as the overall pressure of the air decreases, the pressure exerted by oxygen in that mixture, the so-called partial pressure of oxygen, also decreases. So at sea level, you're breathing 100% of normal sea level oxygen. And if you go up to the highest human habitation, which is in Peru, you're breathing air that has about 50% of the normal oxygen. You go up to base camp at Everest and you're breathing air that has about 45% of the normal level of oxygen. You go up on the top of Everest and you're down into the 30% of normal oxygen level. And the loss of oxygen, or the lack of oxygen, creates a term that we call hypoxia, creates hypoxic conditions. Now, overall, it's not just hypoxia that might be a problem, but overall loss of pressure. And so low pressure is termed hypobaria. So we have combined hypoxia and hypobaria in these high altitude conditions, in these high altitude regions. And that raises some questions about what this does to the animals that live there. All right, just sort of pausing a little bit and going off on a slightly molecular direction. This was work that was done over many years by 3 guys who won the Nobel Prize in 2019 for their work on the hypoxia-inducible factors. How do cells sense what is going on with hypoxic conditions? And essentially what happens is we have this protein here. It's called HIF-alpha, hypoxia-inducible factor alpha. And if you have oxygen present, so you're breathing normal oxygen, this gets destroyed. But if you don't have oxygen present, this survives. It moves into the nucleus. It binds with its partner, a molecule called HIF-beta, forms this dimer, so the two bind together and then turn on the activity of hypoxia response elements. These are genetic sequences in the DNA that act to regulate a series of target genes that do various things within the cell or within the body. And in mammals, certainly, the sort of things that are turned on by these hypoxia-inducible factors in the absence of oxygen are angiogenesis. So you get an increase in the growth of blood vessels into certain regions of the tissue. And this is actually something that's really important in tumors. So when you have a tumor formation, tumors often signal angiogenesis, and so they induce the growth of blood vessels into the tumor to provide them with oxygen and nutrition. Clearly, that's not good for the individual. It's good for the tumor, but not for the individual. So HIF proteins can turn on angiogenesis. They can alter metabolism, with the balance between metabolizing carbohydrates and metabolizing fats, which can affect the amount of oxygen that an animal needs. They can turn on the process of erythropoiesis, which is the production of additional red blood cells. And over time, if you're up in altitude, you're going to find that you're going to have thicker blood. You're going to get more red blood cells growing. But the other thing that the HIF complex does is it turns on the production of more HIF-alpha. And so this is a positive feedback loop. So we can use that positive feedback loop to measure the hypoxic effect of various conditions. So we can put the insects, in this case Drosophila, in various conditions and look to see, do we see an increase in HIF-alpha production? And that's what we were doing in the lab, based upon the notion that as the climate warms up, insects are going to move higher and higher, and they're going to start experiencing unexpected hypoxia. So the questions we asked in the lab were, what are the effects of low oxygen and low pressure on SEMA production? SEMA is the Drosophila name for the HIF-alpha, for the hypoxia-inducible factor alpha, the light blue one. And the second question is, can we separate these effects? Can we tease apart the effects of hypoxia and the effects of hypobaria? And this was a fairly sort of novel bit of work. This hadn't really been done before, so we're quite excited about that. So here's our beast. This is Drosophila, and we keep them in these culture dishes here. They feed on this blue food here, and females on the left, male on the right. Most of the work we did was on females. We did a little work on males, but in general, the males did not seem to display the same set of phenomena as the females. It's almost as though the males were disposable. Once a female has mated, she doesn't need a male anymore. She can keep laying eggs for a couple of weeks after that. So males, we have limited uses in this context. Okay, so how did we do this? What did we do? Well, we simulated high altitude. And our conditions were 24 hours at 18,000 feet, which is just a touch higher than base camp. And the way we did this was we built a Perspex hypobaric chamber, which we attached to a vacuum pump, and we could then evacuate the air out of that chamber. And when we do that, you can see we can measure the altitude using this altimeter here. This is actually my old skydiving altimeter that I repurposed as a laboratory instrument. And so this is real time, so you can see we're evacuating the air at a pretty fast rate. We're already up to 11,000 feet. There's 12,000 feet, 13,000 feet, and so on. And as I say, we took these guys up to 18,000 feet within the chamber. We had 4 treatments. We had a sea level equivalent, and that was basically just putting one of these bottles in the incubator next to the chamber. Normal atmosphere, normal pressure, normal oxygen, so just like breathing at sea level. We put one of the bottles inside the chamber and evacuated the air, so it was now at 18,000 feet, and that's equivalent to the base camp. So it's low oxygen and low pressure. Now we had 2 gas cylinders, one with 10% oxygen. That's about half of normal, which is what you would get at a base camp. And we had one with 42%, which is about twice as the normal rate, or the normal concentration of oxygen in air. And we used these to isolate the factors of hypoxia and hypobaria. So hypoxia, we induced by pumping in 10% oxygen at normal pressure. So that one was sitting outside of the chamber. So that would give 10% normal pressure, but hypoxic. The hyperbaric one, what we did was we pumped into just this bottle here. We had this extra valve coming in there from the tank. We pumped 42% air into 42% oxygen into this bottle at half pressure. So that gives you normal oxygen at half pressure. So these were the treatments we did, and we left these treatments running for 24 hours. There were technical reasons why we couldn't really go beyond 24 hours, which was unfortunate. And then we utilized the central dogma of biology that, information in the form of DNA is transcribed into the form of messenger RNA, which then can be translated into proteins. And what we were doing was we were isolating this messenger RNA. So if a gene was turned on, it would be making its messenger RNA. So we would extract the RNA and then use this machine here. It's called a real-time PCR machine. And we used primers, which is the way of amplifying just the genes that we're looking for. Primers for SEMA, that's our target gene. And we also had primers for what's called tubulin. And tubulin is a gene that is always being made at about the same rate. So we could use that as an internal standard within our experiments. So we could use this, sorry, my lights just went out, a motion sensor, there we go. If you sit still long enough in this university, all the lights go out on you. So what we did was we extracted the RNA and did real-time PCR on using these 2 sets of primers. Real-time PCR you may have heard of during the pandemic. It became the sort of gold standard way of detecting whether somebody was infected or not. Okay, so what results did we get? Well, at sea level, so that's normoxic and normobaric, so normal oxygen, normal pressure, we had low levels of SEMA production, as expected. When we took our flies up to high altitude, so that's hypoxic and hyperbaric, we had an increase in SEMA expression. That was expected. When we isolated hypoxia, what we saw was a huge increase in SEMA production. And when we isolated hypobaria, so that would be normal oxygen but at low pressure, we saw again a large increase in SEMA production. Both of these experimental situations gave more SEMA production than the normal high altitude situation, indicating that there's something else going on in here when you isolate these things. Maybe it's that under normal conditions of high altitude, these 2 interact, feed back against each other, and modulate the level of expression. But either way, this was a novel observation. The notion of a gene being turned on by pressure alone is something that I can't find that in the literature anywhere. So I believe this is the first demonstration of a specific hypobaric gene expression response in any organism. So our two questions: how do insects adapt to lower oxygen hypoxia and lower pressure? And how can we separate the effects of hypoxia and hypobaria? Well, CEMA expression increases. And we can separate them, but there may still be some sort of interaction going on, which it still would need to be elucidated. So conclusions. Conclusions are that as insects get higher and higher, they are likely to experience different and maybe some novel patterns of gene expression. The significance is the expression of these genes is, for an animal exposed to hypoxic conditions, is a good thing. It helps the animal survive under those conditions. So yeah, it's going to make it easier for insects to move higher and higher. And then the other significance, obviously, is that it is a novel observation. As I mentioned earlier, the problem with this is these are short-term experiments, so 24 hours, and we were going to try and set up some long-term experiments at that pyramid research station. We were working on all of the permissions from the Nepali government, and then the pandemic just killed the whole thing. And we're never able to get it going again, which is kind of a shame. Hopefully somebody one day will be able to set those experiments up on a long-term basis. Okay, so this was the part of the talk that looks at the research work, and these are some of the students who worked on this over the year. And these guys presented at NCUR in 2019. Again, the pandemic got in the way of some of the NCUR presentations for several years. All right, so I want to switch gears slightly and talk about cardiorespiratory function in humans. And this is based around the trek up to the base camp. And these are some of the students who were there last time I was up there. Again, you'll remember, the higher you go, the lower your oxygen level. And this is the altitude of Everest Base Camp. And you can see you're going to have about 45% of oxygen that you have at sea level when you're out there. This is the trek. We fly into Lukla at about 9,000 feet and then spend the next 10 days or so getting up to base camp at around 17,500 feet. The altitude profile here shows that we start in Kathmandu, we fly up to Lukla, and then we spend 2 days in a town called Namche. And this is a standard acclimation protocol. So what you do is you stay at the same altitude for 2 nights, but during the day you go up and then you come back down again. We do it again up at Dingboche here before we end up at Gorakshep, which is the highest place we stay, which is a cold and miserable place, to be honest. It's beautiful, but it's not an exciting place to try and be comfortable. So there's the altitude profile. So the question becomes, how does the body respond to that? How does the human body respond to it? It doesn't always respond like this. They're not always smiling like this when they're at altitude, only when there's a camera around. And so what you find is, and we took our pulse oximeters up with us, what we find is if you measure the pulse oximetry and the amount of oxygen bound to the blood at different altitudes, you get an almost perfect mirror image of the altitude profile. The higher up you go, the less oxygen you have bound. And so at high altitudes, you might only have 80% saturation. If this was at sea level, you'd probably call 911 and get somebody on extra oxygen to help them. But at this sort of altitude, it's not uncommon. So if we look at it in a little bit more detail as we go up in altitude, you can see that overall we get a reduction in the saturation, especially at the high altitudes. It seems to be sort of an exponential relationship up there. One of the ways the body compensates is by increasing the pulse rate. So as you go higher and higher, oxygen saturation goes lower and lower. What you need to do is increase your pulse rate so that you can compensate and get more oxygen moving around the body. So again, a resting pulse rate of 95 at sea level would be alarming, especially for somebody who was young and fit. At sea level, you might expect it to be somewhere around about 60, but altitude 95 is about normal. Blood pressure goes up the higher up you go, especially what's called pulse pressure, the difference between your systolic pressure and your diastolic. That's essentially how much pressure the heart is generating in a given beat in order to move blood around. And you can see that takes a large increase in pulse pressure at those higher altitudes. Now, all of these data here were collected on students, and partly to engage them in sort of the physiology of the program, but also so I could keep an eye on them, frankly. So I could spot things with their pulse rates and their blood pressures and their pulse oximetry data that might give me an indication that, okay, this student is perhaps not doing as well as they claim they're doing. So we used this as a way of sort of checking on everybody's well-being. Now, I think if I was to ask people, how do we control respiration? Most people would probably think, oh, well, we measure the oxygen level and we breathe harder if there's not enough oxygen around. And that's partially true. There are chemoreceptors in the blood vessels, the aorta and the carotid artery, that will measure blood oxygen level. And if the oxygen level goes down really low, you'll get an increase in ventilation. The normal oxygen level is around here. And it really doesn't vary very much under normal circumstances. But if you go below this hypoxic threshold indicated by the arrow, you'll start to see an increase. We actually measure CO2 levels in our blood and regulate ventilation based upon the level of CO2. So small changes in CO2 level can have big changes in ventilation rate. And this all results from these cells down here in the medulla oblongata of the brain. So we typically measure CO2 levels unless oxygen levels fall too low. So how do we do that? Well, we have these chemosensitive neurons in the medulla. And if we have a decrease in breathing or an increase in exercise, we produce more carbon dioxide, fairly standard. That enters the brain across the blood-brain barrier, and through this chemistry here, makes the cerebrospinal fluid a little more acidic. That is sensed by these chemosensitive neurons. You get an increase in breathing to compensate. It's a natural, negative feedback system. And when you have a decrease in CO2 level, because you've breathed more or you exercise less, the blood will become slightly, or the cerebrospinal fluid will become slightly more basic, and that will decrease the rate of breathing. So we have this balance that's mostly regulated by CO2 levels, not oxygen levels. But at altitude, we get this battle between the two. So low oxygen availability triggers these chemoreceptors in the blood vessels to stimulate ventilation. So you start breathing more. And as you breathe more, what you're doing is you're breathing in more air and flushing out CO2 from the lungs, which lowers the CO2 level in the blood, which then leads the sensors in the medulla to shut down breathing. So you stop breathing. And then a few moments later, the oxygen sensors kick back in and they make you breathe more. So you end up going back and forward with this sort of metabolic battle between hyperventilating and hypoventilating. And it's really quite uncomfortable, actually, especially if you're trying to sleep, because all of a sudden you'll be breathing away, and then you'll stop breathing for 10, 15 seconds. And then you start breathing again. And you end up with this battle that makes it really difficult to get comfortable. It's called Cheyne-Stokes breathing. Fortunately, with a little bit of time, the brain can reset itself by pumping bicarbonate into the cerebrospinal fluid that traps hydrogen ions in the cerebrospinal fluid, and that allows the body to increase the rate of breathing despite the fact that CO2 in the blood is going down. And this takes a few days in order to happen. So yeah, this is how we would regulate ventilation. Now, another way to do it is what's called pressure breathing. And this was an experiment I did at Lobochet, that of the COVID fame. And essentially what you do here is you breathe in and you purse your lips as you do it. So a little video there. One I recorded earlier. And so what that does is it increases the intrathoracic pressure, which increases the pressure of oxygen in the lungs, which increases pressure of CO2, and allows you to hyperventilate without messing up your CO2 levels. And you can see we started off here with a saturation, I think, of 73% or 76%. And by pressure breathing for a minute or so, I've got my pressure, my oxygen saturation up to 96%, which is actually very close to where it is right now. I tend to be slightly under 100% with mine. So this pressure breathing is a really useful technique, and climbers use it. I was actually taught this technique by a Navy SEAL back in my skydiving days, and that they were trained how to do this so they could go up much higher than everybody else, and it works. A lot of climbers use it. It looks kind of funny and it sounds kind of funny when you're doing it, so you tend not to do it in polite company. Okay, so that part of the talk there was really based upon work that I did on the Go program to Nepal, this Sherpa Life and Culture program. The last piece I want to talk about is more of a lab exercise, and it looks at what's called the diving reflex in humans. We don't have a great one, but we do have a diving reflex. So diving mammals, there's really sort of three groups of diving mammals. There's humans, cetaceans, which is whales and porpoises and dolphins and things, and pinnipeds, which are seals and sea lions and the like. This is a humpback whale. Goes by the name of MN1304581. It's a photograph I took last year in Antarctica, and there's an organization called Happy Whale, so you can take a photograph, you can upload the photograph to the Happy Whale website, and the markings on the bottom side of the fluke and the little sort of indentations in the back of the fluke here are like fingerprints. So they are unique, and you can identify a particular whale. This was the first time this whale had been spotted. It's subsequently been found up off around the Galapagos earlier in the summer, which is kind of fun. You can track your own personal whale. So thinking about diving animals, diving mammals, different whales, different seals are able to do this with quite remarkable abilities. Cuvier's beaked whale here can dive to almost 10,000 feet, or has been seen to dive to about 10,000 feet and stay underwater for about 3 hours. Humpback, 700 feet for 30 minutes. The elephant seal here, this is a juvenile elephant seal making friends with some gentoo penguins here. I don't know what he was doing on top of that hill, but there he was. 5,000 feet, 2 hours of a dive. Weddell seals, which are really quite common up there, 45-minute dive to over 1,000 feet. So this is remarkable when you consider how lousy humans are at this sort of diving, with the exception of a few people who do free diving. All right, so what strategies can an animal use to maximize dive duration? There's really three: enhanced oxygen stores, redistribution of blood flow, and a cardiac dive reflex. So just going through these three briefly. Enhanced oxygen stores. Here's the human over here, and we can store a certain amount of oxygen within our body. Some of it we store in the lungs. There's always air within our lungs that has oxygen in. We store some of it in our blood, bound to the hemoglobin within our blood, and then we store some of it bound to a protein called myoglobin in our muscles. But overall, we're not great at this. We can't store that much oxygen. And you compare that to the Weddell seal or the hooded seal here, they typically don't store much in their lungs. They actually empty their lungs before they dive, but they have large amounts of hemoglobin storage in the blood and large amounts of myoglobin storage in the muscles. The color of seal and whale meat is really, really dark red, and that's because of all the myoglobin that's in the blood and in the muscle that can then bind the oxygen. So these enhanced oxygen stores are used to keep the animal alive while it's spending large amounts of time underwater. That would only work to an extent if the blood flow distribution throughout the animal stayed the same. It doesn't. There is a redistribution of oxygen stores within the body. So the muscles, as I said a moment ago, they have lots and lots of myoglobin in them. And so they can store oxygen, and essentially they get cut off from all blood flow. And so if you look at this angiogram from a harbor seal, this is pretty old data, but you can see these blood vessels on the surface are open. And when the animal dives, these blood vessels vasoconstrict and shut down and restrict blood flow to the muscles, which means that the blood that has all that carbon, that has all that hemoglobin with the oxygen bound to it, is reserved for three organs: the heart, the lungs, and the brain. You lose any one of those three, you're dead. you can survive for a long time as long as you keep those three organs going. Muscles can go all hypoxic and it really doesn't matter that much. So they're quite tolerant to this. So what these animals do is they redistribute blood flow. We're not very good at that. The last piece is what's called the cardiac diving reflex or dive reflex. And again, this is nice. I really like old papers which have, the original data in them. And so this is a set of data from a harbor seal, and it's based on a sonogram. And so what you have is on the surface, you can see lots and lots of heartbeats, occurring fairly high frequency, maybe a couple of beats per second. There's a time scale down the bottom. And at this point here, the animal dived. And all of a sudden, you can see the heartbeats spread out, maybe 10, maybe 12 seconds apart in some cases. So the dive reflex results in a drastically lowered heart rate. As the blood is being reserved for the heart, the lungs, and the brain, the heart rate itself can slow down because it doesn't have to pump as much blood around. And these two things together, the redistribution of the blood flow and the cardiac dive reflex, are really good at maximizing the time of a dive, allowing these animals to spend large amounts of time underwater. So the question then is, this is the one we were approaching in the research lab or in the teaching lab, do humans display a cardiac dive response? And the answer is yes. And what you can do to demonstrate this is to immerse the face. There are sensors around our face that will detect water. And when they detect water, they do a couple of things. Number one, they inhibit breathing. Well, that makes sense. You don't want to be breathing with your face underwater, which is why some people have a hard time snorkeling, because the face is underwater, the receptors are being triggered, and yet you're still expecting to be able to breathe. You can train yourself to get past that. And then the other thing is, it's, at least in whales and seals and the like, it slows down the heart rate. And the other question is, are there temperature effects? Do you see a greater dive reflex at colder temperatures than you do at warmer temperatures? So these are some of my students in the lab, and what I would do, this is one of my favorite labs. I love doing this, largely because I don't stick my face in cold water, but they do. And you can see the temperature over here. It's pretty chilly. So what we do with this, we hook the students up to EKG monitors, and in some cases, we will measure their ability to continue to breathe normally through these snorkels. In other cases, we leave the snorkels out. And we try this at different temperatures to see what effects these different temperatures have upon the cardiac rate, to see what effects they have on this human dive reflex. All right, we're not seals, but nonetheless, if we look at the EKG data here, this is the sort of a blow up of one of the EKG peaks, PQRST, atrial depolarization, ventricular depolarization, ventricular repolarization. This looks a pretty normal EKG profile, and I just grabbed it off one of these peaks up here. But what you can see is prior to the dive, which occurred at this point, we had a fairly high frequency of EKGs, i.e. 60 beats per minute of the heart, and the EKG amplitude was relatively high, 0.81 millivolts, indicating the amount of electrical activity that the heart is generating. Well, the student, and I can't remember which student this data comes from, stuck the face underwater, and you can see almost immediately two things happen. The heart rate slows down, and the amplitude, in other words, the muscular activity of the heart, decreases, goes from 0.81 millivolts down to 0.54 millivolts. And then the moment the student surfaces, brings their face out of the water, you can see we go back to normal breathing, and we have a slightly increased heart rate. It hasn't quite got up to the full resting there. There's still a residual dive reflex, and we can see the amplitude is recovering as well. So yeah, humans do have a dive reflex. It's just not as good as that of a whale or a seal. So, which is why we don't, I don't know, swim underwater and eat krill and things like that, because we couldn't do it very well. Now, one part of our lives when we do have a good dive reflex is as infants. So typically a baby under about 6 months has a really powerful dive reflex. So this little girl here, looks pretty happy underwater. She is very young, and she's been put in water. She's totally immersed, and her dive reflex is slowing down her heart, so-called bradycardia, and it's inhibiting her breathing. So she won't breathe while she's underwater unless she's left under too long, which naturally a parent wouldn't do. Eyes are typically wide open, as maybe as some sort of alert response, which is quite reasonable. And so babies are really good at this. After about 6 months, the dive reflex and all of these other responses tend to wane. So by the time you get to, my age, retiring and all that sort of stuff, the dive reflex is pretty minimal. So what have we done? We've talked about, I believe, 3 instances of hypoxia. We've looked at what happens with gene expression in insects and under hypoxic conditions. We have looked at what happens to humans with regard to changes in pulse rate and oxygen saturations at higher altitudes. And we've looked to see what happens in terms of dive reflexes allowing animals to withstand the hypoxic conditions of being underwater. Now, just to sort of leave you with, I'm not going to say it's an uplifting end message, but there's some interesting theories going around about what's going to happen to oxygen levels in the future. And if we look at oxygen levels at the moment, what we're seeing is they're gradually decreasing in the atmosphere. And this is partly due to loss of vegetation, habitat destruction, fossil fuels, increasing temperatures, increasing humidities and the like. And so we have this gradual decrease in oxygen concentration within the atmosphere. And you can look at this and you think, maybe is that sort of beginning to sort of be a parabola? Is it beginning to sort of decrease more rapidly? And if we extrapolate that out, we end up with the rather disturbing conclusion that in 3,600 years, the oxygen concentration in the atmosphere at sea level will be what it is at base camp at Mount Everest. And in about 5,000 years, the oxygen concentration in the atmosphere will be very close to zero. Life as we know it, if this is true, if this modeling is true here, is going to have to make some real changes. They're going to have to be great drastic changes in terms of the ability to withstand hypoxic conditions. So we won't be around to see this. It would be very interesting if we were, but nonetheless, I think it's an interesting sort of future projection. And not all of it is anthropogenic. I mean, there's some sort of basic geology involved in this too. So I'll leave you here. This is one of my favorite views of Mount Everest. I took it a couple of years ago now from a neighboring valley. So this is Everest up here with the south summit, the Hillary Step, and the summit over there. This is Lhotse peeking out from behind the ridge, and this is Nuptse up here. So with that, I'm happy to entertain questions. Rick, if you can let me see if there are any, that would be great. Rick has been monitoring this whole thing. So thank you. Let's see this, hang on a second. There we go.
Hi.
Hi.
I'm just curious. I'm an alumnus of Susquehanna, and I'm just curious. I've been doing some studying of environmental science since, and I'm just curious. Do you see any evidence in what you were studying over there that, like, for instance, with the fruit flies, do you think there's suspicion that they might evolve a little bit and some evolution take place to help counteract those conditions? Or what do you suspect with the idea of evolution with those items and with other insects and mammals?
Well, I think you're onto something there. And this is one of the reasons why we wanted to do the long-term studies, because insects, Drosophila especially, you can get 30, 35 generations a year. And what we've found in other studies is they actually can evolve really quickly. So within 10, 20, 30 generations, you can get substantive changes in the morphology of the insect. So absolutely, I would expect that were we able to do those long-term experiments, we would see more evolutionary changes occurring. And that raises the perhaps uncomfortable notion that these natural populations are going to be able to evolve to manage and cope with these higher elevations as climate change makes more habitat available to them. That's a great question.
Thank you so much. I appreciate it.
You're welcome. Actually, I'm just looking at the list of who's online right now, and I see there's at least one person who was on one of the, or two. There's two people as I look at the list of who's online. I see some of my old students. It's very nice to see you guys.
Professor, there's one question that was typed in. It asks,
Okay.
are you following up on the fly work?
Alas, I don't think I'm going to be able to, as Kathy noted earlier, and I'm retiring, and I'm going to lose my lab, or have really lost my lab right now. I'm moving over to Scotland, where I hope to be able to continue to do some work in the sort of the field of high altitude physiology, but more on a sort of a writing level rather than a practical level. So I'm hoping somebody sometime is going to stumble across this and want to sort of pursue it a little further. I would love to have done it, but it's the pandemic and the advancing ravages of age have got in the way.
And I have another typed in question. What are you looking most forward to in retirement?
I'm going to flip that around and say what I'm not looking forward to. What I'm not looking forward to is not having the contact with students. I think that's been one of the things I've really enjoyed over the past. I worked in North Carolina, taught there as well. So 35 years of teaching and being surrounded by young people the whole time is a privilege. I mean, it's just been absolutely brilliant. And I really will miss that. I won't miss the grading. I'll tell you that. I absolutely detest grading stuff, but nonetheless, yeah. I'm looking forward to being able to relax a little, to have time to write, have time to engage in a few other hobbies. I like photography. Virtually every photograph I showed today was my photographs. I'd like to do that a little bit more. And so we'll just have to see. I'm looking forward to the beer as well. The beer in Scotland is better than the beer in the US, I'm afraid.
I have another typed in question. In Bolivia, women at high altitude are required to go to lower altitudes when pregnant. Anything like that in Nepal?
No, they don't tend to do that, not to the same extent. But I think overall there are more people living at higher altitude in places like Bolivia and Peru than there are in Nepal. The populations at the higher altitudes in Nepal are relatively small. So north of that town of Lukla that I showed you on the map, there's probably only about 3,000 people who live permanently north of there. And so it's a low number. So a few of them will go down, but you know, the Himalayan population has evolved, and I think that is the right word, to manage those high altitudes. The populations in the Andes have done the same, but interestingly, they've done it in different ways. So different populations have evolved at high altitude in different parts of the world to manage their high altitude, their hypoxia, but in different ways. Sorry, my light just went off a bit. So yeah, in Nepal, no, they typically don't head down to lower altitudes.
There's a Matthew online here who has his hand raised.
Hey, Matt. You're mute. Oh, there you go.
Hi, Dr. Richard. I don't have a question, but I just wanted to say thanks for letting me relive some memories of COVID on the mountain.
You're very welcome.
Wish you a happy retirement.
Thank you. Matt was one of the students who was on the last trip up to base camp.
I have a question. Have you experimented with Diamox in human subjects?
I have, and I typically take Diamox when I'm at high altitude. Diamox is a drug. It's a carbonic anhydrase inhibitor, and what it does is it essentially traps protons, hydrogen ions in the bloodstream. It causes you to pee out a lot of bicarbonate ions, and that can be problematic because you can become dehydrated. But as long as you watch your water intake, it's actually a really nice way of allowing the body to keep hyperventilating to get the oxygen that you need. It's sort of a quick way to induce the same sort of responses I went through in the medulla there. Yeah, Diamox is really handy, and most of the students who are up there, if they're not allergic to sulfur drugs, most of them will take Diamox as well. It makes a big difference. It allows you to acclimate more quickly.
Another question here. How are populations in Nepal that rely on ecotourism preparing for the expected changes in the climate to come?
Yeah, it's interesting. In some ways they're not. They're not as they're not following climate change to the same extent that we do, despite the fact that they live it much more than we do. A lot of the operators over there are continuing to do the same sorts of things. I know climbing Everest is going to become quite different in 20, 30 years, simply because of the loss of snow. It's actually going to become much harder in many ways. It might be a little safer because you're not going to have the avalanches, but climbing on rock is harder to do in many ways than climbing on smooth snow. And so, they will continue to market and sell their natural resource, which is a unique resource. The high Himalayas are a wonderful thing, and I encourage everybody to try and see them at some stage in your lives. But yeah, it's going to be interesting to see how they do it. Quite a few of the operators have gone out of business. The pandemic nailed a few of them. Some commercial decisions in this country by operators here hurt some of the operators out there, which was a shame. I'm not naming any names of which companies it was, but yeah, it's going to be different over the next few years. It's not going to be the same. Tim, you have your hand up.
Hello.
Hey, Tim.
Yeah. I just want to ask, because I know that in our set of research in 2022, we did have that confusion with the results. I was wondering if this most recent year helped clarify any of those results, or if it just added more to the jumble.
You mean the dive reflex?
No, with the flies.
Oh, sorry. Yes, with the flies. No, I think we sorted some of that out. I think there was some sort of technical issues with the PCR that were going on then that I think we managed to sort out. So I'm pretty comfortable with the results as they stand right now.
Wonderful.
Yeah.
I do not see any more questions online.
Oh, Jeff King just raised his hand. Last one, Jeff. You're muted.
There we go. You can hear me now.
I can.
Yes.
How are you doing, Jeff?
Not bad. Thanks for the revisit to animal physiology, the purse breathing and partial pressures and all that. Yeah, I still remember that. And yeah, just nice to hear that and enjoy retirement. And Leslie Imhoff says hi. I had lunch with her today.
Oh, very nice. Yeah, very good. Thank you.
Thank you.
All right. So thank you all. This was fun. I enjoyed putting all this together. It was kind of fun to put everything together in a different context and to be able to not necessarily deal with all of the detail, just the bits of detail I wanted to talk about. So thank you for the opportunity. I hope you enjoyed it, and I wish you all a very good evening.